So thank you for watching the Young Adults group. We've got June here, who's the Director of Research at the Society, and also Robert, who's been in a lot of the trials in the past, and they're here to share an update on the pipeline for the next couple of years, trials that either are ongoing or we expect may come on board, and then also to, on Robert's part, share about his experience in the prior trials.
Yeah, I'm June Kinoshita. I'm the Senior Director of Research and Education at the FSHD Society. I joined the Society back in 2012. And at that time, the genetic cause of FSHD had been identified. And when I looked into it, I thought this was an amazing drug target because basically FSHD is caused by the expression of this gene called DUX4. We all have it, but in adults, it's suppressed. It doesn't get expressed, turned into protein and have any kind of biological activity. But in FSHD, it does get expressed when it's not supposed to, and it gets expressed in muscle, and it's very toxic to the muscle. So the reason I thought this was an amazing drug target is if you can knock it down, and we can talk about all the different ways you can knock down a gene, that there really shouldn't be any serious side effects because it's not doing something else to keep a person alive, keep the system going. It's supposed to be suppressed. So I was very excited about that and wanted to be part of this effort to really develop treatments around it. In some ways, developing ideas for treating it was the easy part of getting treatments to patients. I think the much more challenging part is that FSHD, as most of you listening here know, is really variable from one person to another. The age that the symptoms start to appear, where the symptoms appear, how fast or slow it progresses, it's very unpredictable. So when you have a clinical trial, you have a limited amount of time, 6 months or 12 months is on the long end, during which the company running the trial has to somehow prove that their drug has made an impact, that people treated are better off than people who are not treated. Well, when you look at 20 people with FSHD in your test group and 20 people in your placebo group, how are you going to tell? Like, they're all so different, right? So the challenges are like, can you figure out which, pick 20 people who are going to progress kind of in a similar way during that next, 12 months, or that you hope they will, statistically. And how are they going to change? Is it through change in their speed of walking, or their bicep strength, or core strength? There's something else, and you have to have a really good way to measure that and to be able to detect what may be a very small change, a few percent, 5%, 10%. And it's got to be robust enough that it's not part of the noise of the natural variability that you're going to see in this group of people. So those are the big challenges, and the society has been funding research to try to develop those trial outcomes. I think one of the most important things we did was to really work closely with Robbie Tawil and Jeff Statland to establish what's called the Clinical Trial Research Network. And that is a growing network of FSHD centers across the US and now in Europe and in Canada and even beyond, that work together like a center without walls. Because, with a rare condition like FSHD, like even a big center like Kennedy Krieger, where people come from around the world, to go there, and they have a large number of patients that they work with, they would not be able to recruit enough people for, anything beyond like a phase 1 trial. So to do a phase 2 and certainly phase 3, you need multiple centers. So So establishing this network was a big milestone. That was done in 2016. And I'm really excited to say there are now over 30 centers in this network internationally. So anyway, I'm going slightly off off topic here. I just wanted to kind of introduce a little bit of my interest, what was so promising about FSHD, some of the background, the kind of research, and the kind of what we call infrastructure work, building kind of the highways to make all of this flow, has really led us to this moment now where we have multiple clinical trials, more on the way. And we actually face the challenge of we may not be able to recruit enough patients fast enough in certain locations, and we have to be very strategic about, how are we going to keep things going so that we don't have trial failures.
That's
And
helpful context. And Robert, did you want to share some of your experience in the prior trials?
Sure, I can give you knowledge. And first, I'd like to thank you, of course, and June and the Society for all the work that's been done over the years. And it's a privilege for me to be participating in this discussion, as well as the other trials and things of that nature that I've participated in. I can talk a little bit about my little personal journey and how it's taken me through these trials and what have you. And I can do that. My name is Robert. It's called Robert Rowe. My name is actually Robert Rosania, but my handle online is Robert Rowe. This is Victoria. She's my significant other and yeah, caretaker. She's walking this journey with me, and it's wonderful to have a supportive partner who is able to, work with me to overcome the challenges that we all face with this condition, obviously, as it progresses. And it's just been a wonderful partnership. Insofar as my history, I began, I guess, as an adolescent, right? So probably in my teens, where I realized that I couldn't do things in gym that other kids could do. Okay, so I'm just going back sort of like to the beginning. I couldn't do pull-ups. I couldn't do rope climbs in gym class, and I felt inferior to the other students. It's like, why couldn't I do that? When I got into high school, I would do canoeing or kayaking, pardon me. And there's a technique called an Eskimo roll, which is when you are upside down in the water. There's a maneuver that you can do with your shoulders to push yourself and flip yourself back around. I can never do that either. So I just had inherent weakness in my upper body. Now my legs and my lower torso, all that was very strong, but my arms or my shoulders, I just had weakness, and we never knew. It was undiagnosed. Nobody knew what FSHD was in the 80s or 70s, right? Nothing like that. So, other manifestations were collapsed rib cage, what they call pectoralis excavatum. My pectors and my pectoralis, they're not really that large. I have winging, the classic symptoms now that we all understand with FSHD, angel wings, wing of the shoulder, and inherent weak upper body. Now, that's just something I lived with for years. But where it really came to a head for me was probably at around 2009, Zach, when I was building a home. And things that I was doing, I found a little bit more challenging, like climbing up on some scaffolding. For some reason, I just felt I was having a little bit more difficulty. Or if I took a bicycle ride, I felt that I had difficulty pedaling up the hill, and it never really occurred. Nothing really. It's like something was wrong. I just didn't understand it. And then when I was walking, every once in a while, I felt like my left leg would sort of get like spaghetti and feel like it was almost like it was going to give out, like somebody hit you behind your knee, and then you sort of fall. And so I ultimately went to see a doctor, a neurologist, which was recommended, and they suspected that I had a muscular dystrophy. Now, this is 2009, let's say. So I was already 30, 2009? I was 45 years old. And to have somebody tell me that I have muscular dystrophy, I couldn't believe it. I mean, I'm thinking of Jerry's kids, you know, Duchenne, the children, God forbid, that are in the wheelchairs. I'm like, I have muscular dystrophy. But so what did we do? I mean, like anybody going through something new, you know, there's a lot of fear and uncertainty. And I thank my father, who was very good at researching, came across the society, the FSHD Society, and he reached out, and then he had me call, and God bless Daniel Perez. He was very accessible when we spoke. Just an absolute wonderful guy. I can't speak well enough about Daniel Perez. Just a wonderful guy. And the thing he said to me, Bob, is to get into research. That's where you know, help this participate, be part of the solution. And, you know, given that I was close to, you know, Philly, closer to Baltimore, he had recommended Kennedy Krieger, right? Which is part of like Johns Hopkins and Dr. Catherine Wagner's group. So now I should just prelude that with that I actually did get a genetic test at University of Pennsylvania from a department of neurology before I went to Kennedy Krieger. And they were kind enough to pick up the cost of the test. I think the MDA picked it up, provided that I allowed the data to be shared of the results of my study. So they actually covered the cost of the test, which was, you know, not inexpensive. And then once I got that formal diagnosis that it was FSH, that's when I reached out to Dr. Wagner's group, and they took us in intake, and we brought my whole family. So I have a brother and a sister and a mother and a father, right? So they wanted to see, since it's an autosomal dominant type genetic trait, they wanted to see what parent it could have come through and if any of my brothers or sisters had the disease. And so we did some tests, muscle biopsies. We did another genetic test to confirm, and it was determined that my brother has FSH and my mother had FSH. It actually came on my mother's side. Interestingly enough, my mother was extremely, very mildly affected. I mean, I don't think Dr. Wagner was able to look at her face and thought she saw some asymmetry in her face and felt that she, you know, was the one before she had the genetic confirmation. But my mother was in the 80s at the time, and she had no mobility issues, right? But I was the proban, right? That's what they call that, I believe, where I have the most severe presentation, most clinically significant presentation of the disease. And my brother, mildly affected, has no lower strength issues, maybe upper arm slightly, but nothing like me, right? So we went to Kennedy Krieger.
Your sister had none, right?
What?
Your sister had no.
No, my sister and my father, she was completely free of the disease, right? And one thing that really made an impact, and I've said this before, if you've ever seen me on any of my other, you know, videos or what have you, is, you know, like anything, you go through, you can maybe get angry, you question why. It's like, of all the things, why me? And, you know, why this, that, and the other. And you can get frustrated and angry at life, right? In the situation. But the thing that really had an impact on me was when we came out of that elevator on the 4th floor at Kennedy Krieger. Anybody who's done that trip will know what I'm talking about. You come out of the elevator and you see children in wheelchairs. I mean, children, babies, young children, and their parents. And when I walked out of that elevator, I instantly felt like I was cured. I mean, I was a lucky one. Yeah, it's unfortunate that we have these conditions. And that really inspired me to want to participate in every possible thing that I could do. Any study, any muscle biopsy, any blood work, any test, anything I would be happy to do. And that's my attitude in all these trials right now, is I really, for me personally, I do these trials for the future, for others primarily. It's not a self thing. I mean,
Right.
whether it happens for me or not is a bonus, but and so that's how I look at these trials. And some of the trials, and I can get into some of these to get specific, you or natural history studies, the Resolve study, which is an important study. June, maybe you want to talk on that a little bit, but that's where they looked at my progression over a year or two, right? And there's no drug. So it's not like there's not going to be an immediate payback. Well, I'm going to take a drug and I have a chance to be cured or treated. You're putting in the hard effort of building a road to build a bridge to the future, right? And that requires, natural history studies, which all that data that they collect, observing your strength, your mobility, your functional abilities, any other clinical biomarkers, MRI studies, anything like that over a period of time will help pharmaceutical companies who actually do engage in these trials, because it gives them some kind of baseline, right? Since, June had mentioned the variability of this disease, it makes it difficult because it presents in everybody differently at different times in different ways. So it's really difficult. How do you measure that? So it's good to get a large baseline of participants to, as like June said, you get enough data that you get through that noise that could potentially be, if you don't have enough of a size, you know what I mean, of a cohort of tests. So I'm sort of jumping around, but I'll just go from my history if we're okay. I think the first trial was the Wellstone 58, which was back in 2014. I'm not exactly sure what that was. It wasn't a drug study, Zach, but it was more observational, I think, and measurements, maybe MRI, maybe blood, maybe strength, maybe I think could have been 6-minute walk tests, and those types of things. So that was the first real study that I guess I participated in. Dr. Doris Lung, who had worked with Dr. Wagner, developed or was working on an MRA study, which, allowed them to look at the active disease within the body, either through inflammation, right, where there could be DUX4, inflammatory markers, there could be fat infiltration, when the muscle gets replaced with fat, and you can actually image it and actually see the effects of either the act of the disease or the consequence of the disease, right? So that was very, very important to help. And now Fulcrum, we're doing the Fulcrum trial now, the loss by mid-mod or Redux 4. I've been in that trial since, I'm going on how many years now? 4 years almost, 3 years. Yeah, I think I was the second recruit at the Dr. Wagner's group for Redux 4, Fulcrum. And that was a double-blind, placebo-controlled study. And for 6 months, we didn't know if we were on the real drug or the placebo. But then COVID came, right? And that required the extension, the changing of the dynamics of the study. So the study went further. And to Fulcrum's credit, they went into what's called an open label extension. Are you familiar with that is? Right. Okay. So now everybody after that 6 months of the trial, placebo-controlled type double blind, can get onto the real drug, right? So now I've been on the real drug now for probably 3 years or so. And now they've gone to phase 3, as you probably know, right? And they're recruiting and currently enrolling and engaged in that study. And I am continuing on with the Fulcrum trial. What other studies have I participated in? Boy, I've been in a lot. I did the A-tire study. This was one of the earlier studies. June, you may be aware or familiar with A-tire. Can't hear you. Okay, you're shaking your head. I was excluded
Yes,
from
of
that
course
study. Oh, yeah, okay. It's been a while. I was excluded from that study because I had what was called a Jo-1 antibody that was detected, which was an exclusionary condition, and so I wasn't able to do that test. Another test I participated in, it wasn't at Kennedy, it just happened to be at University of Pennsylvania, was the ACE-83 trial, which was a myostatin inhibitor, right? And this was an injection. So what they did is they looked at your muscles with MRI and they determined a target. And in my case, it was the tibialis anterior, which is the muscle that runs down the front of your, like, what is it, tibia?
Mhm.
In your leg. And they did these injections. Now, it really wasn't a pleasant thing, because getting injections right there on the front of your shin there, there's not much room for a needle to go in. It was guided, I think, but it was pretty unpleasant. But the idea there was, with myostatin inhibition, the idea was that it was going to make the muscle, they saw evidence of the muscle density or mass increasing, okay? But one of the questions that I had about that was, well, if the muscle making factory is defective in that DUX4 is expressed in that process of a myoblast becoming a myotube, you know, those things forming a muscle, if that process is destructed by, you know, DUX4 or an inflammatory response to DUX4, you know, throwing gasoline on to make more, you know, on a fire to make more muscle, can that really make a difference if the underlying muscle making is defective? The answer really wasn't known, right? But I think the conclusion from that test was that while they did observe an increase in mass, right, that there was an increase in the size of the muscle, let's say potentially, that there was no functional improvement to the muscle. So the increase in the muscle mass did not result in an increase, let's say, in strength. So that's where that test ultimately ended up. Now, I guess there was some good potential information that came out of that test, but that study then was concluded, to my knowledge.
Yeah, if I
Which
can
then,
make a
yep,
quick comment about the Acceleron study. So they saw an average 15% increase in muscle volume for people who were on ACE83. And the functional, sorry, my rooster's crowing. The functional outcomes were hard to interpret because it was a 6-month long trial. And what they found was that people on the placebo were also improving, like, because everybody was doing the same set of tests and assessments. And there's something called the practice effect, that in the beginning, you know, you start to learn how to do these, you get better, you might have a little, you know, you show improvement. And that effect really only starts to separate from a real drug effect after maybe 6 months, a period of time. So one thing we always wonder with the Acceleron trial is maybe they stopped it too soon, because we know from the Fulcrum Resolves, the Redux 4 study, that through the sheer accident of it happening right during COVID,
right.
they doubled the length of the trial. It was originally supposed to be 6 months long, like the ACE83 trial. But because the clinics were closed and they couldn't go, you know, they said, okay, let's extend it to 48 weeks. If they had stopped collecting data at 6 months, they would not have seen an effect. It took a year before they saw significant difference on things like reachable workspace. So I think one thing everybody needs to understand is, in a disease like FSHD, even as they do clinical trials, they are on the bleeding edge of research. They are taking their best guess at how we measure the effect, but they're actually doing, you know, the study itself is kind of conducting the research to see if that outcome measure is going to help detect it. And if you guess wrong, if you guess that 6 months would be enough time and it wasn't, you end up having to put a drug on a shelf that may, if you had run it for 12 months, who knows, it might have had some benefit. So so I just want you know, our listeners to understand that, you know, because I often, you know, and I totally understand, I hear people saying like, what's taking so long to get a treatment? I think, like, we only figured out the gene like 13 years ago. This is blazing fast speed when you think of the effort that's involved, the kind, you know, the careful experiments that have to be done to develop treatments, make sure they're safe, and then get them into people and set up the whole infrastructure to be able to run trials. So So I just want to, and somebody has to be out there on the leading edge, right? This generation of people with FSHD, you are the first to have any hope of some kind of treatment. And it may feel like it takes a really long time, but somebody's got to be out first, and you are it. So I just want people to understand that, and to understand how incredibly important it is to do that research as rigorously as you can, and not cut corners, and have a group of patients that, yes, I'm sorry, we can't mix in people in wheelchairs, because then how are we going to compare, 3 people in a
Right.
wheelchair with some people who are walking? How do we interpret that? Um, so anyway, I just wanted to mention that when you're talking about the Acceleron trial, that
Sure.
we
And
learned
I've
a
had
lot from
this. I've had this discussion with a number of the researchers, and one of the things that I think about, June, with this disease is it's a somewhat obviously it's a complicated disease. Many diseases, I guess, are, but there is the active disease, and then there's the result. Okay, and the result is fatty infiltration. And the unfortunate thing, and I sort of think of like that gentleman who had, poor young kid who had the Lorenzo's oil,
Mhm.
where he had the myelin, uh, delamination or whatever it was called. It was deterioration, which caused it was the myelin that was deteriorating, which then caused the nerves to be exposed, and then they got damaged. And even when they got the treatment to stop that demyelinization, the damage for those who already went through it, the nerves were already damaged. So yeah, you could stop active disease. And I equate that kind of situation to FSH in my mind, because, like June was saying, if you're in a wheelchair, you have severe disability, and you've lost the majority of your muscle, right? And you mostly now have fat replacement. How do you build muscle on that? You really, currently it isn't. So my expectation is, June, and I could be entirely wrong, but with a disease like a fulcrum, or a treatment like a fulcrum or avidity, or any of these other the treatments that could potentially come online, could stop the active disease from continuing to progress. But then you have to look at the people who have been so affected by this disease, that have the fat that's left. How do you recover and restore muscle to that area? Because I guess you need muscle to build on top of muscle, right? Muscle
Well, you need
damages, right? It repairs,
you,
and then it gets bigger, right?
you need a store of satellite cells. Those are what we call the muscle stem cells. So these are the kind of little baby cells that are not really fully muscle yet, but they have the ability to, if they get the right signal, they will mature into muscle fibers and so on. So in most of the tissues in our body, there's a repository of stem cells that are just waiting there. And let's say there's an injury or something, you lose them, there's a signal that says, hey, well, I need to recruit some of you guys to come and turn into, you know, skin cells, or, liver or whatever. And so same with muscle. What we don't know is if there's still that capacity to regenerate in people who have experienced, this a lot of deterioration in a muscle. We really don't know. And conservatively, you might say, well, maybe that muscle might not have any regenerative capacity left, but we really won't know until we try, right? And until you stop the DUX4 process, maybe there are a few cells in there that can, now that DUX4 isn't in the picture, they're able to regenerate. So that's one thing I'll just mention.
Sure.
Other is
And
that
maybe there could be a cocktail.
well, there are people working on treatments to replace those missing satellite cells, or to
Wonderful.
transplant muscle precursor cells that can grow into muscle. So and I was astounded to learn this year, there are 2 companies doing this that are starting to seriously develop this for treatment of FSHD. One of them is Vita Therapeutics, spun out of Johns Hopkins, actually building on research that the society funded a number of years ago,
Nice.
where you can take, I think it's usually they use skin cells because skin is easy. It's not as painful as getting a muscle biopsy, but also you can reprogram them. This is one of the great innovation discoveries in modern biology, is that you can take a cell that has already matured and kind of put it in a time machine and make it go backwards to a more primitive state, into this kind of stem cell-like state. So these are called induced pluripotent stem cells. And you can keep them, put them on ice and keep them for a while. And then when you're ready to use them, you in theory, you transplant them in, put them in a test tube, add the right factors to have it start turn into, muscle or whatever. And then you, at the right moment, inject those into the muscle of somebody with FSHD and hope that, the idea is then they will kind of take up residence in that muscle, mature into healthy muscle fibers and replace what was lost to the disease. That is the idea. Vita is using cells from people with FSHD. They can treat it in the test tube to shut down the DUX4, basically kind of do like genetic engineering in those cells. So now you can put them back in the person, and these corrected cells will not express DUX4. It should grow into healthy muscle. And they came from that patient originally. So hopefully the immune system will not reject them, because that is the big, that's a big factor in any kind of cell or organ transplant, is that your immune system will say, oh, this is a foreign body, I'm going to go attack it and get rid of it, right? So, and then you have to take immunosuppressive drugs, which then now you're a little vulnerable because your immune system is shut down, and you could, you've got to be super careful not to catch an infection or anything like that. So if you use your own cells, it's called an autologous cell transplant. You won't have to take these immunosuppressive drugs. So that is the idea with Vita Therapeutics, and they're already, we put out a recruitment notice for them earlier this spring. They were looking for people to volunteer to contribute some biospecimens so that they can grow up some FSHD sample cells and kind of start.
Wow. Is that the kind of thing they're going to take a little like EpiPen type thing, and they just take a little piece
a little
of
skin punch, yeah. But
okay, that may be something.
yeah, it's really, it's kind of, even a few years ago felt, sound like sci-fi. It still sounds like sci-fi, but people are doing it.
I'll mention that. I'll sign me up for it. I guess I'd be happy to give that. Definitely.
And the other person who's working on this is Rita Perlingero at University of Minnesota. So she's taking a different approach, which is to collect cells from people without FSHD, just on a healthy, regular, normal, control, or, and you can culture the cells. And then what you do is you would transplant that, but you would have to immunosuppress that patient. And the rationale there's a much more abundant supply of people who can, of these, genetically normal iPSCs, this induced pluripotent cells. So you would be able to provide them much more at much less expensively. You can just get a batch, and everybody, who needs something can. Whereas Vita, it's a custom solution. It's your cells. It probably takes half a year or more to grow up those cells and divide them up and get them in big enough numbers. It's like having a whole scientific laboratory devoted just to developing a custom treatment for you. So you can imagine the cost of that will be quite high. But it's pretty amazing that this is now being translated into what a real therapy. It's not sci-fi anymore. And then of
And
course,
and I think the pace, as we see, I mean, 2014 is when we started to see these clinical trials potentially take some form. And look at that, we've already got what, 4 or 5 already that's been done, and now we've got 3 or 4 now, and then we have another 4 potential. So, and it's just going to continue to build. And one of the things I'll say to people who are thinking about a trial, participating or whatever, or get frustrated, every trial in my mind, June, is a success, or Zach is a success, because it's like Thomas Edison, and you asked him how he made a light bulb. Well, he had, it took him 1,000 light bulbs to make a light bulb, 999 failures. So even the failures move the knowledge forward. So you're always failing forward, in effect. And then you have your successes as well. And it's like a snowball effect. And I'm very optimistic, that we're going to see some real viable treatment and or cure, hopefully within the next 5 years, I should think. I'm hopeful. We'll just see.
And I will just say those studies that did not have a positive result, I don't want to call them failures, because if it were done well, that is a successful experiment. It answered the question you set out to answer, which is, does this work? And if it didn't work, okay, that's disappointing. But if it was really well done, you can move on. You're not arguing about, well, you didn't design it right, and did it, I've seen areas of science where poorly done experiments just slow everything down. And so every one of them, you're absolutely right. Everyone is valuable as long as it's done with rigor. And I also wanted to mention, you talked about the natural history studies. So there is the big study that's going on right now. It's called MOVE, Motor Outcomes for, I can't remember the whole, Jeff Statland spins out these amazing acronyms all the time. But, and the idea is that these are evaluations that can be done in a doctor's office without elaborate equipment, and can be used at your annual appointment to show, measure and detect change over time. That is the sort of idea, because when we actually have treatments out in the world, you're going to have some way of tracking all of the thousands of patients out there who are going to be on treatments. And so he's thinking ahead, like, what's a practical tool that will let us do that? But there's another study, it's a sub-study, or kind of an expansion, or a spin out from MOVE, called the MOVE Plus. And this is a really important study, and for some reason they are having a really hard time recruiting people for it. Maybe because everybody wants to go right into clinical trial. Maybe that has something to do with it. But this MOVE Plus is really important. What it is you do all the strength assessments, the same things you do in MOVE, but you also get a whole body MRI. And you also you provide a muscle biopsy, a little needle biopsy. And why this is important is, ideally in future clinical trials, you want people to be able to go into an MRI, you go in and look like, oh, this muscle here is starting to be fatty infiltrated. Here are the muscles that are affected. 6 months later, 12 months later, you want to take another MRI and go like, wow, look at that. That muscle hasn't changed, or it's even gotten better or something, right? You want to see the change. Right now, you could take MRIs and make claims, and the FDA will go like, prove that change in that MRI image actually means that this person has gotten better, is stronger, is able to walk faster, is able to lift a weight they couldn't lift before. If you just had the MRI and none of this other measurements, the FDA is not going to buy it, right? And So you have to validate the MRI as a way of tracking disease progression. If you can do that, it will simplify the clinical trials. It might shorten them. It might be back to, well, at 6 months now, we can see something that we know is going to be meaningful to that person's function. And
Right.
the FDA will accept that. And now we can have a trial in 6 months. It costs half as much as a 12-month trial, or well, maybe not quite, it doesn't scale exactly, but significantly, tens of millions of dollars less than a 12-month long trial. It can mean, it can take away a lot of the uncertainty of, as like one day you go in to do an assessment and like, you couldn't sleep the night before, so you're tired and you're not, it's like there's so many human variables that are less likely to affect what an MRI looks like, because being out on the town one night is not going to dramatically change how your MRI looks, whereas it can dramatically change how fast you're going to be able to walk 10 meters or something, right? So and then the biopsy is the same thing. If we can collect a teeny sliver of muscle and show that the DUX4 is no longer active in that muscle, and the MRI that goes with it shows that the muscle is not getting fatty infiltrated, these are very compelling forms of proving, the activity and efficacy of a drug. So they are kind of the holy grail for FSHD clinical trials. So we need that MOVE plus study to get there. And
So
right
let me
now,
ask you.
nobody's signing up for it. So I just want to put it
Okay.
there. It's
Let,
really
let me ask you that, because let's say, with the move, because we did the resolve, which obviously then led to the move.
Yeah.
But our move does not have the uh, difficulty of enrolling people. They've got a sufficient, or they have a good number of people who have enrolled in move in and of itself.
MOVE itself has enrolled well. They have close to enough people. Some locations can still take a few more.
Yeah.
But the MOVE Plus has been very slow to recruit. It's a
And I
big,
assume everybody who's in move has the option to do the MRI study and or the muscle biopsy, right? To
they have
become
the
part of the
option.
plus, but they're declining?
They're just not enrolling in it. I mean, it is more demanding, and you've got to lie in an MRI for a couple hours. And I can tell you that's not too bad, actually. I think you're allowed to sleep. I
I
did an
sleep all
MRI
the
study
time
where
in
I was
those
not
things.
allowed to sleep for 2 hours, and that was torture.
No, I sleep in all those, sometimes they're 90 minutes, but maybe some people don't feel comfortable in a tight space. I mean, to me, like you say, I'll just fall asleep and
Yeah.
relax in it. It's the muscle biopsy. I've had, like, now June, probably 4 biopsies, and Fulcrum took a couple, and that was a little dicey because one hit a nerve when it was a guided, or it wasn't it was an unguided muscle biopsy. So she's sort of doing it in the blind in the skin, and that hurt.
Oh,
There was one biopsy that I was numb for about a month on my left side of my foot. It did resolve, thankfully, but I've given a few, and this one here for the xenograft, I mean, this was pretty invasive. This, was under
oh yeah, the
surgical procedure.
xenograph is a big one. Yeah.
Yeah, and then I was like skinned like a fish on a table there. It wasn't pleasant, but uh yeah, I don't know if it's the muscle biopsy that people are pushing back on versus the MRI. Must they go in hand? You must do both the MRI and the biopsy for
Yeah,
both
because
validations?
you do, because if you just look at the MRI, it's like, what are you looking at? What's going on in that muscle? Nobody knows. So I think FDA has a pretty high standard for evidence. And if you just present MRI images and you can't tell them, you have no data saying this is what's happening in those muscles, it's, you don't have a very good, it's not a good biomarker without that added.
Well, can I ask, if I can ask a question, short of a muscle biopsy, and would this only be one muscle biopsy in one target that's picked? Or are these multiples over a period of time to see if
I
there's an improvement?
I think it is, I don't quote me on this. It's a series, but I mean, they're trying to minimize the amount that they do it because they know it's invasive and it's uncomfortable. It's over time. So MRI, biopsy, and then 6 months later or whatever, another one. They've tried to pick a muscle that's still pretty healthy, so that they don't want to worsen a muscle, or harm a muscle that's already kind of on the edge or whatever. So they got to find people who have just the right kind of
Right.
but it's
Yeah, and couldn't
contribute.
they, again, I'm just, if I may, theorize here for a moment. Short of the muscle biopsy, let's just for a moment assume that that is one of the things that people are resistant to, and I can relate to that, being on the receiving side of muscle biopsies. Couldn't one do, let's say on a certain date, you get a whole body MRI, and let's say you're looking at active disease in your bicep, let's say on the right arm. Let's just pick one in my case, for example, and you're seeing there are things going on there. And during that time, you're getting strength tests or functionality tests or reachability, like workspace, any kind of measurement, strength, mobility. And then 6 months later, just to use your kind of time frame, another MRI look, if it's dosed, if you're on a medication or something and you want to see if there's an improvement, you take a picture of that area, you see if the MRI suggests an improvement, and then you correlate that, imaging to the strength tests or functional tests or dexterity tests that relate to that muscle. And couldn't that be sufficient? Because, one of the things that I could say, well, let's think about this. When you take muscle and you cut it up, you're injuring that muscle. And if you're going after a muscle that's already unhealthy because of disease, and you're going to go in and even do more damage to it by taking muscle away, it's like, is that really the best thing to do? You're going after a muscle that's already sick, and you're taking
It's
more of it out.
it's not ideal. I think I should mention they're also taking blood samples.
Okay.
So the real holy grail is if we can find something in blood that correlates with what we're seeing in the biopsy, like we see a change, like we're seeing a whole flood of things that happens when DUX4 is turned on, and then and you are able to detect that in blood. And then when the drug works and that stuff stops, and you look at the blood and all those signals are gone or down, that is the holy grail, because it's not as invasive. You're not taking muscle from someone, you're just collecting it from the blood. But it is, it's searching for that proverbial needle in a haystack right now.
Right.
We've supported research using very advanced, very sensitive techniques like mass spectroscopy. It's way beyond just doing stuff in a chemical tube. It's physics and all this stuff to try to find these rare biomarker molecules that we hope are out there. If we can do that, that would be, that would really change things.
Sure.
But it's possible that if, with that biopsy data to back it up, that the MRI on its own, the FDA might accept it and say that is an we believe that this is showing, shown us what it shows, and we'll accept that. So, but right now, it's like, we don't have time to waste. We've got to do the best possible experiment to try to find the best possible clinical trial outcomes, for the future. Even as we're already doing clinical trials right now, because we know, they've got to be more in the future. We may have better compounds. Right now, losmapimod looks really promising. Maybe there's a super losmapimod out there, that will, or these mRNA drugs that are, being tested, and then the gene therapies and all, there's going to be a lot of things coming along. I suspect that the ideal treatment is going to be a combination, depending on how advanced you are and what is it, so on. So, and all of this, the right now is the critical time to kind of get the science, to get those biomarkers and imaging markers really nailed down, because the long-term payoff will be tremendous.
You all have shared a lot of helpful information. I know people will be interested as well in hearing about the next 12 to 24 months. Any highlights on the near-term pipeline that you two would like to share?
June?
Okay, me. Well, I just want to mention a few things that are out there already. So we haven't talked about the Maneuver trial, which is from Hoffmann La Roche, major pharmaceutical company. This is kind of similar to the ACE83, this myostatin inhibitor, but it's supposed to be much more kind of have a better dynamics inside the muscle. It'll be around longer. And they learned from Acceleron. This is going to be a 1-year trial, not a 6-month trial. But it is a very, very demanding trial. It's got MRI, it's got biopsy, it's got, it's a long day in the clinic.
Right.
So I have some concerns, and they're competing for patients who, for the, I guess, the Avidity trial now, because the Fulcrum phase 3 trials fully enrolled now. So they're competing with Avidity. And I just want people to, and Avidity sounds really exciting. It's very cutting edge, mRNA, and, could stop the underlying cause of FSHD. That's really important. I can definitely see why people want to be in that trial. But Maneuver is, I think it's important to support that approach to treatment too, because I hear from a lot of people like, I would love to grow back some muscle. I'd love to grow back some muscle. Well, we want that to be part of the offerings, the treatment possibilities that are out there for the future. So, and of course, if you're in the Maneuver trial now, you can take other the losmapimod or, whatever else, if it gets approved, you will have access to those treatments. Right now, it's really, all hands on deck to help move forward as many therapies as we can, because it's going to be that, the combination, the more options we have, the more successful treatments there will be for everybody.
That maneuver, the Roche myostatin inhibitor, is that also June, an injectable localized to a particular target muscle, or are these?
No, I believe it's systemic. I believe it's an infusion, goes into your bloodstream, goes all over the place, and let me just double check that. I don't want to be
Interesting,
but I don't get that wrong.
because I got to tell you, ACE was a little challenging because I think I got 5 or 6 injections, and that was just one muscle. And I'm like, well, my goodness, how many muscles are affected? How many injections would I need? It would be worse than a rabies shot, of course. But when you're looking at muscle, I think I have to remember what there was a drug that came before ACE83, and it was a GYM or Jim something. I don't remember the code for that drug.
Okay,
But
yeah.
you got to watch, right? Because a muscle is the heart is a muscle, right? So, what happens if you take something to encourage muscle growth, and then the side of that is that there are other muscles that really shouldn't be targets that could be affected, right? In a systemic approach.
Yeah, so I stand corrected. It is a subcutaneous injection, which means just goes under the skin and then it kind of diffuses from there. So it's not directed to the muscle. So it's not one of these deep intramuscular injections. So it's less
Okay.
invasive.
Right.
And with a treatment for FSHD, you want to reach a bunch of, I mean, I don't know if they inject it close to muscle that they know is affected, but as you know, like at the end of the day, there might be muscles all over the body in different places
Mhm.
that are affected. So I think I think the idea here is the off. Yeah, I don't know. It's interesting because there could be effects on muscles that are currently healthy. I mean, they'll get even bigger.
Yeah, yeah, I guess that's gonna be a
Yeah.
phase 1 thing. How about Arrowhead Pharma? They have ARO DUX4,
Yep.
right? Is that for
Yes,
FSHD1?
that's right. They have just launched or about to launch a phase 1/2 study in New Zealand. And my understanding is that New Zealand has, they have very good research. They have a small FSHD community. They have a registry. And I think the government makes it a little bit easier to get approval for that type of a study with an experimental therapy. So my understanding is that was one of the reasons they decided to start it in New Zealand, get the important safety data and stuff there. But if it looks promising, I'm pretty sure the phase 2 will probably be, I mean, it's going to need a lot of sites. So I think there's,
Mm.
it'll be in other places.
And do
Maybe-
you know, is that also an mRNA treatment or
Yes,
an
it
injectable? It
is.
is.
There are 3 companies, Avidity, Dyne, and Arrowhead, are all using these mRNA kind of antibody conjugated oligonucleotides. So they have this little bit of DNA or RNA that's complementary to DUX4 RNA. So it'll find it, kind of lock onto it, but you have to get it inside the muscle. So they use an antibody that targets the surface of the muscle and helps carries the therapeutic molecule, the mRNA, inside the muscle cell. So they all have that same basic strategy, but devil's in the details, like how well it penetrates and how well it binds to them could vary depending on whatever each company has decided to, how they've built their molecule. Miracule is another one, is also using
That's the other one, Miracle,
yeah,
right?
Miracule. They're partnering with Sanofi, so that's exciting because that's another giant pharmaceutical company that will bring its resources to the clinical trial. So
How
lots
about
happening.
if I
Epic,
can? Oh, go ahead,
yeah, Epic
please.
Bio is another one. This is the one that's using kind of CRISPR type gene editing technology to silence DUX4, and they are using adeno-associated virus. So they're using a virus to carry their therapeutic payload into cells. It'll be the first therapy of that type to go into clinical trials for FSHD, and they're applying to start a phase 1 trial, I think, or it might be a 1/2, but by the end of 2024, next year. So
If I can bring up other ones like that, at least that I've come across, in the educational side of things, right? The academic side, Scott Harper, I think he also had an AAV approach of some sort.
yeah,
Chris Hamida, I think with Jones Lab has been working on, is it CRISPR-Cas9 or something to that effect? Do you
that's
know where those two sit in the spectrum or arc of, a pathway potentially into any kind of clinical trial? Do you,
yes.
connect?
So Scott Harper's technology has been licensed to a company called Armatus. Um, which is still, I think, what we call doing preclinical IND enabling studies, which is the data you need to the package you need to present to the FDA for them to approve it to go into, into human trial. So, Renogenics is the name of the company that Peter Jones's group has started from their work, again, using gene editing technology. So I think they're also doing what I would call these, these pre-IND or IND enabling studies. So, definitely stay tuned. The race is on there. I think they're all working like crazy.
So, I know we're getting close to time. I did think it'd be helpful. So we've got Fulcrum. I think they're supposed to report results around September of 24. And then we've got, as you mentioned, a number of others as well. Do you think, based on the difficulties you mentioned with kind of measuring progression and how effective the treatments are, do you think if these treatments are effective that the measurement processes are at a place where they'd be able to pick that up?
We hope so. So let's talk about the REACH trial. That's Fulcrum's phase 3 trial. So they will complete data collection. The last patient will have completed the trial in next September, as you said. It'll probably take them 3 to 6 months to analyze the data and pull out, whether there is a significant difference between the placebo group and the drug group that's on the therapy. And what I found really exciting about losmapimod is during the phase 2 trial, which is really still looking for safety primarily and tolerability, they were looking at some clinical outcomes, and there were a couple that jumped out that actually showed a difference. And one of those was called reachable workspace. And this is this thing where you hold this device and you're kind of moving your upper body, your arms, through different quadrants of space. And they saw a significant difference between the placebo group and the test group. So that was amazing. You're not planning to necessarily be able to show that in phase 2. So fingers crossed that they'll see the same kind of effect in phase 3, but you never know. You might have had, it might have been a fluke. So that's one thing. I think they also, people on these surveys, when they described how they were feeling during the trial and things like that, they saw a significant difference in some of those quality of life, I think, patient-reported surveys. So one of the challenges here is, how do you persuade the FDA that a 15% difference, or like that person, these patients on losmapimod did not show a loss of reachable workspace, whereas the placebo group all, on average lost 15% of their movement, or whatever, the volume of space they're able to move their arms in. Well, what does that mean to patients? Does it mean that more people are able to still keep brushing their own teeth, or taking a shower, or able to work on a computer? What does it actually mean? What does that 15% loss mean to the quality of life of the person? So there's work going on right now. Dr. Han, who invented reachable workspace, is trying to do that kind of validation study. There is all of you who are on our mailing list will have gotten an invitation to participate in a survey that has that aim to try to do an additional study to kind of map reachable workspace to patient relevant, patient-centered outcomes. So if you got that email from us, please do the survey. Please try to participate in it because it's critically important. And so that's, do you have any other questions? I'm happy to answer.
Yeah, I think we touched on a lot of them, right? So far as what's potentially coming down, Zach.
I did have one follow-up actually. So you mentioned in phase 2 they may not necessarily be expecting to see efficacy. However, I think a lot of people like in the society like associated with it will be looking for indications. Can you give an idea of what the changes in a phase 3 versus a phase 2 that might lead to clearer revelations of efficacy?
So, I mean, the fulcrum is looking at using the same measurements. So what we're looking for is there's a lot more people doing it. So much more significant, like just it's a statistics thing, right? So instead of 72 patients, 36 on the drug and 36 placebo, I think there's 240, so 120 people in each group, or maybe something like that. So just looking at that many more people should give you a much stronger, statistically stronger result if it's real, that's,
That makes
Yeah,
sense.
Zach, and I think one of the challenges, because we've discussed this amongst ourselves quite a bit, actually, is now because I've been on this Fulcrum trial now for almost, I can't believe it, 4 years, 3 years? It's been a long time. And I was certainly on the open label extension or on the real drug for at least 2. So, the question is, how do you know? And June sort of touched on it. What constitutes or what qualifies as yes, it's working or it's not working? Well, June touched on reachable workspace, and I'll just say some anecdotal experiences from my own. I think when I started doing the reachable workspace, and that's where you're like sitting in a chair, just to explain this to people what that is. You're sitting in a very uncomfortable chair, by the way. It is not the most comfortable chair. And you're sitting like this, if you can see this. And then you have a computer with an imaging that takes this graphic of your body, right? And there's like quadrants to touch on the space that Jim was referring to, the actual space, so to speak. And then you do motions like that, like how far, like you see, I'm limited without bending my elbow. How far can I lift it? How far can I lift this? How on an angle this and so on? How far can I go back? These are the motions that we're looking at. And they do that. And then that's the reachable side. And then they put on like a 3-pound weight, right? On your wrist, and you repeat those movements, and the computer tracks them, right? Now, when they measure you, they have to measure your ability to, excuse me, to reach 90 degrees. And based on some of those motions when they set up the test, they can determine whether you're a high functional or a low functional. If you're a low functional, the reachable workspace movements are less. There are less of them. If you're high functional, thank you. If you're high functional, you do more motion, more movements, more measurements. And in my personal case, when I started Fulcrum, I was borderline. Sometimes I would make a high, and then other times I would make a low. So I was like right on this threshold of between a high functional and low functional. And I think over time, again, does it mean something? Hopefully, that I was consistently in the high functional. Because I'm just one person, okay? One person. I can't really say for sure. But is that a success? Also, the disease, is it a success if it's enough to slow the progression of the disease? So, if over 4 years I've lost, let's just say, to put a number on it, hypothetically, I'm not saying I did, but let's just say 5%, I can't lift 5%, or I lost 5% in a strength test of a particular muscle, okay? On the drug. Does that mean that it's not working because I lost 5%? No, it could mean that it is working, because then if I weren't on the drug, could I have lost 10%,
Exactly.
15%, 20%? And my heart goes out to the people at Fulcrum or anybody else on these trials, because as June said at the outset of this, it's a very tricky disease. It's not like, okay, I see a blood level, your cholesterol is this, and now you take a drug and now it's lower. You reduced your cholesterol. It's not like that. It's a lot of interplay, different progressions, different muscle groups. It's a lot of moving pieces, right, June? How do
Mhm.
you, especially with a small group, and 230 is still small. I mean, compared to like 20,000 on a study, right? Now you got some real data to work with. So it's tough to say, but I'm optimistic that it's doing something, right? And I don't think, with a placebo, there's always that placebo effect. And that's why I sort of took the questionnaires with a grain of salt. Because I actually saw a comment, just to give you a specific example, with the ACE-83 trial that I tried, the injecting at one muscle group, and somebody felt online, they made a post about, well, I just have seen improvements across the board. And I think that was a placebo effect. I think that was a wishful thinking of an individual, and maybe that manifests itself some improvements. People maybe push themselves subconsciously to do a little bit more, or work a little harder and think they're going to see an outcome. So it's really difficult to get a subjective assessment. I mean, it's always nice to have, but I take all that with a grain of salt. I ultimately always tend to go back to what are the real measurable, quantifiable things. Strength tests, range of motion tests, functional workspace. Can you do this? Can you do your, close your jacket up okay? Or can you touch the back of your head? Can you do this? Can you reach your pocket? Can you do this? These are the different movements at Fulcrum. And, that's the kind of thing you look at, I think, to help validate, to determine if a drug is efficacious or not, right?
Yep. Very well put.
I appreciate both of you taking the time tonight.
Our
It was our
pleasure. Thank
pleasure.
you for having us.
And again, I can't thank June and the FSHD Society enough for everything they've done. And as I say, I bring up Daniel Perez all the time. I talk about. He's just a wonderful person, and he's just been a great guy to have on this effort for all of our sakes. And the amount of work, if I could just touch on one thing for Daniel Perez, we are really far down the line here. I know, as June said, people want treatment. When is it going to be here? When is it going to be? We have come so far so quick. But without the early work of the society and Daniel Perez, who had to start to get go to Congress, right? He would have to write up these and make presentations to all of that heavy lifting was done with the likes of Daniel Perez and the society. And without that foundational work, none of this other could have happened. And it's like you're building a road. You got to excavate the area first, right?
Mhm.
And he did all of that, and the society did all that work. So, in how many years? 15 or so short years.
Yeah, well, Dan started 30 plus years ago.
30.
It's totally, there was nothing, he just really, and a couple of other people. I mean, there was no society, there's no email list like hey, everybody, like, where are the patients? Who are they? Where are they? How am I going to find them? Who are the doctors that see them? Where's a researcher who might be, could be persuaded to work on this rare disease that they never heard of? Just from nothing really built what we have today. So we truly stand on the shoulders of, giants who had the faith to kind of stick it out for years and decades to get
Right.
us where we can now, launch.
They're the real pioneers, Zach, of this whole effort. And for anybody who's watching, the first thing would be, well, is there a treatment? What is it? Get involved. You can go through a disease and be angry at the world. You can be frustrated with the things that you don't have, or you could take it. And I like to think of taking lemon and making lemonade with it. Take this and make something positive, and that is get involved, subscribe like to the society, get involved, get in patient advocacy, get with groups, certainly get onto the clinical trial research network that's out there. Maybe you can put some links to that. And I think it's important. One of the things that helped me get so involved with the research is that you develop a history with a group. And even though Dr. Wagner's not there, it's now Doris Long, they've got all of the history they have with me. And, they can look at all the different trials. So, if you're going to get involved, and I recommend it, I can't recommend enough to get involved in research, get a relationship with the group that you're going to work with, because when another trial comes on and you're there, even if you're finished another one, they have your experience and they have the history with you. It's much easier for you to get into another trial. So get that engagement. June, I'm sure there are links on the society's page to people in geographic areas, for centers in that area for which to engage, and just start that process, as I did so many years ago.
Yeah. Or if you can't, it's under our research tab, Clinical Trial Research Network, but also you can email navigator@fshdsociety.org, and you'll be connected to a member of our team who can help you, connect to the study or clinical trial that you want to be part of. And I will also say, just to reiterate, every person makes a difference in a disease like FSHD. Every person who gets involved makes it. You're not just like a little data point that's not going to change. Every person matters. And to to extend your analogy about making lemonade from lemons, there ain't a lot of lemons on the tree. You can't just say, okay, we're gonna get gallons of lemonade, but there's not that many lemons, and you can't just let them sit on the tree and rot and not be of use. So anyway, that's obviously it's a big decision, a very personal decision to get involved, but I just want people to realize, if they're thinking about it, it really makes a difference. It really moves the ball forward. So
All right.
Well, thank you, Zach.
Yeah, right.
And we'll be back. We're happy to come back anytime to talk more about it. We love talking about this stuff.
Indeed.
And thank you, Robert. It's a pleasure to see you.
Likewise, I look forward to seeing